Te hei Māori ora, ina mana o tēnei whenua, ina waka o nai tā e whā, te whare e tūnei, tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko Ngāti Pākehā toku iwi, ko Greg Cook toku ingoa. Good evening, everyone. My name is Greg Cook, and I'm the Deputy Vice-Chancellor of Research and Innovation. It's a great honour to be here tonight to celebrate research excellence and present the Distinguished Research Medal and the Carl Smith Medal to two of Otago's finest researchers, Professor Peter Fineran and Associate Professor Andrew Reynolds. Research excellence is not achieved in isolation. It is cultivated within a scholarly environment that prioritises intellectual curiosity, interdisciplinary collaboration, and the courage to pursue challenging questions, values that are integral to the University of Otago's academic culture. Across the sciences, humanities, health sciences, and the arts, our researchers advance knowledge, challenge established assumptions, and contribute insights of significance not only to Aotearoa New Zealand, but to the global academic community. As we face global challenges from climate change to public health, from cultural resilience to technological transformation, research excellence is not a luxury, it is a necessity. Universities like the University of Otago are beacons of evidence, creativity, and integrity in a world that needs them more than ever. So here tonight, we've come to present our two research medals. So I'm going to tell you a little bit about them, and then we're going to hear from the recipients. So the Distinguished Research Medal is the University of Otago's highest research honour. It recognises exceptional, world-class research, long-term contributions to the research community, meaningful impact on academic fields or wider communities, and dedicated service to the university. The purpose of the medal is to highlight and celebrate researchers who have achieved outstanding scholarly excellence and have strengthened the university's research culture. It serves both to honour individual achievement and to promote a standard of research that advances knowledge and benefits the wider community. I would now like to welcome Professor Julia Horsfield to introduce Professor Peter Fineran as our 19th recipient of the Distinguished Research Medal.
Kia ora, Greg. Tēnā koutou katoa, no mai haere mai ki te kaupapa o te wā, ko Julia Horsfield tōku ingoa, ko o te tumuaki o te kaupeka Biomedical Sciences, nō re rā, no mai haere mai. It gives me great pleasure this evening to introduce the 2025 winner of the University of Otago's Distinguished Research Medal, Professor Peter Fineran. Professor Fineran is a world-leading microbiologist, celebrated for his groundbreaking work on bacterial immune systems, particularly CRISPR-Cas. Some of you might have heard of it. Yes. His research has transformed our understanding of how bacteria defend themselves against viruses, and these are viruses known as bacteriophages, and also how bacteria defend themselves against mobile genetic elements. So CRISPR-Cas9 systems act as adaptive immune mechanisms in bacteria, functioning like molecular scissors to cut invading genetic material. And Professor Fineran's group has made major contributions to understanding this field. And this includes the regulation of CRISPR-Cas9 systems, how bacteria decide when to activate these defenses, and how multiple CRISPR systems are coordinated within a single cell. His group also researches mechanisms of genome targeting, revealing how CRISPR can reshape bacterial genomes by removing pathogenicity islands or mobile elements. Peter's group also researches the bacteriophage arms race, uncovering how phages evolve anti-CRISPR proteins to evade bacterial immunity and how bacteria counter these strategies. And lastly, Peter's group has recently researched applications in biotechnology and agriculture, leveraging the CRISPR insights to develop phage-based solutions, for example, for controlling plant pathogens. His work has been published in the top-tier journals like Nature, Science, Nature Microbiology, and include landmark papers on CRISPR adaptation and regulations. These discoveries have not only advanced fundamental microbiology, but also informed genome editing technologies and phage therapy approaches. So Peter's many achievements are very well known, and for this lecture, I wanted to say something in particular that exemplifies the kind of science that he and his team does. And the true story this, I just happened across Peter's latest Nature paper in my literature scrolling one evening, and so I dived in for a bit of a close look because I wanted to know what the fuss was all about. So I'm going to tell you what I found in that Nature paper and why I think this type of research is so exciting. So the 2024 Nature paper from Peter's group was about a particular anti-CRISPR associated protein called ACA. So ACA, together with the anti-CRISPR proteins called ACRs, make up a collection of ammunition that bacteriophages have as part of their attack system to combat CRISPR defense systems that the bacteria have. So when phages replicate really quickly inside a host bacterium, they made a huge amount of this protein, the ACRs, or the anti-CRISPR proteins. And unless these are regulated, the ACRs can build up to toxic levels that then repress all gene expression in the bacteria in the host cell, so much so that this can compromise the viability of the host cell, which is not in the virus's best interest. So this Nature paper by Peter's group showed that these associated proteins, the ACAs, help to reverse ACR toxicity by using a particular domain called the helix-turn-helix domain that can bind the ACR gene promoter and prevent its transcription. But what's really cool is that the same protein domain can also bind the messenger RNA that emerges from the locus and dampen that down and prevent translation of that messenger RNA into protein. So it's like a double whammy protein that hits this ACR gene locus. So in this way, the ACA proteins even out potentially over-enthusiastic anti-CRISPR activity that the phage ACRs do. So the poor hapless bacterium can survive well enough to host the replicating virus and still not be able to kill them. So I thought that was pretty cool, and the work revealed for the first time that these helix-turn-helix domains can be multifunctional and hit both DNA and RNA at the same time. And it also revealed an evolutionary conserved genetic circuit for simultaneous transcription translation control in the phage versus bacterium arms race. So I'll go back on piste now and finish up by saying that Peter's impact from his outstanding work, like this work, is reflected in well over 15,000 citations, an H-index above 60, and recognition as in the top 1% most cited researchers globally. He is a fellow of the Royal Society Te Āpārangi and recipient of prestigious awards such as the Fleming Prize from the UK Society of Microbiology, Rutherford Discovery Fellowship, and now a James Cook Research Fellowship. His leadership and mentoring and collaboration has built international networks and trained the next generation of scientists in CRISPR biology. Without further ado, please join with me in welcoming Professor Peter Fineran to deliver the University of Otago's Distinguished Research Medal for 2025.
Ena mana, ena reo, era karatanga, maha, no mai haere mai, tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko Tohino Korekio te Moanga, ko Waikirikiri te Awa, ko Peter Fineran tōku ingoa, no reira, tēnā koutou, tēnā tātou katoa. So welcome everybody, warm Pacific greetings. Thanks so much for coming along, and thanks for the very kind introduction, Professor Horsfeld. And I'm very glad that you described that work in elaborate detail, because I'm not going to talk about that at all today. So you've actually just extended my talk, which is fantastic. So I also just want to welcome as well everyone who's joining online and also who might be watching this in the future. So let's see if we can get this to advance. Right, so yeah, tonight I'm going to talk about something that I'm really passionate about, and I've been passionate about for a long time, and that is bacteriophages. And hopefully I'm going to take you on a journey so that you can see how fundamental understanding of bacteriophages can also have potential applications in the future. And what we really need to do to get there is to work together as a team. And I may be the one up here giving this talk, but I sit in my office normally, and there's huge number of staff and students and collaborators who I've had the pleasure of working with over the years who have really done all the work that I'm going to present. And I won't have the opportunity to thank them all individually, but just wanted to thank them collectively. Also had fantastic collaborators both within the university and also globally as well, who've made this a fantastic field to work in. I also wanted to do a quick shout out as well to all of the other staff that actually make research possible. We have research happening within our labs, within our departments, but without the other support that happens within the departments and the university, research would be completely impossible. So I just want to thank all of these people. So there was a little virus that happened a few years ago, and now everyone in the world knows what viruses are. So that's made a little bit of my introduction easier. But what you might not be aware of is that every domain of life is infected by viruses. And as Professor Horsfeld said, there are viruses that infect bacteria, and these are the single-celled microorganisms. These viruses that infect bacteria are called bacteriophages, and this is essentially a bacteria eater, if you were to translate it from the Greek, which describes very nicely what they do. And in fact, there's this false-colored electron micrograph image, which I think really beautifully shows you what bacteriophages do. They bind, these shown in green here, they bind to their bacterial pathogens or their bacterial targets. Then they replicate, taking over those resources, produce many more viruses, and then they'll burst out of the cell, destroying that bacterium. And you can see this really beautifully shown here. Now, since their discovery more than 100 years ago, people also thought, well, these could be potential antimicrobials. And in fact, there's been interest in them since then, but obviously antibiotics were highly successful, so they sort of stayed in the background. But more recently, with the rise of antimicrobial resistance and also for looking for other more sustainable uses of phages, people have returned to them as a potential antimicrobial. So I'm going to cheat a little bit here, which is we've had the real pleasure of working with Te Tera Whakamātaki, which is the Māori Biosecurity Network, as part of an MB program that I'm currently involved in. And they put together a very short 2-minute video which gives the background of what phages are, why we could be interested in them, and what applications they may have. And I'm just going to save my voice for 2 minutes and play it to you.
Every year, our world is threatened with many bacteria affecting our health, our natural environment, and food systems. Instead of using chemicals or antibiotics to combat these bacteria, research shows there's another solution, one that's already all around us. In fact, they're the most abundant entity on the planet, more than all other animals, bugs, and bacteria combined. Bacteriophages, or phages for short, are found in soil, water, plants, animals, and even inside our bodies and on our skin. Phages can be thought of as the viruses for bacteria, helping to keep your body and the world in balance. Normally when we think of viruses, it's not good. However, phages can actually be used to treat bacterial infections that cause harm to humans, plants, or animals. They can even be effective on those that are resistant to antibiotics and agri-chemicals. Unlike antibiotics, which kill both good and bad bacteria, phages can be chosen to seek out and bind to the bad bacteria only. The phage works by infecting the bad bacteria, killing it off and leaving good bacteria to thrive. Phages are naturally occurring and biodegradable, a part of our natural world. We've lived alongside phages our whole life. Now scientists are proposing we use them to help protect our plants, animals, and people, to restore our drinking water, rivers, and lakes, or realign our body's health barriers to help heal illnesses and disease. These tiny helpers can play a vital role in maintaining the balance in our ecosystems and provide an environmentally friendly way to combat bacteria.
Well, that sounds pretty amazing, right? So you'd be saying, why are we not doing this already? And this is because it's complicated. And this is because bacteria have actually been evolving alongside their phages for many, probably billions of years, in fact, and they've evolved these immune mechanisms that protect them from phage infection. So much like our immune mechanisms that stop viral infection, bacteria have their own immune mechanisms. So whereas we would be wanting to use the phages to attack the bacteria, these bacteria actually have ways that they can fight back. And this is not a new idea. In fact, the discovery of restriction of viral or phage infection dates back to about the 1950s or 1960s, and this led to restriction modification enzymes, which some of you may have heard of and even worked with in the lab. But in fact, the abundance and diversity of these systems has really become apparent in the last 20 years or so, with CRISPR first being discovered and really rebirthing this area of interest, and then over the last 10 years, even more diverse immune systems being discovered. And so tonight I'm going to mainly focus on the CRISPR-Cas systems, where we've focused most of our effort in the past 10, 15 years or so. So you can think of CRISPR-Cas as the adaptive immune system within bacteria. This is essentially like our adaptive immune system. It has a memory of past viral invaders, stores that memory, and then uses that as a sequence-specific way of seeking them out and destroying them. And so for the non-scientists, this is a picture of a bacterial cell cartoon cutaway, and it essentially has an immune memory bank, which is the CRISPR array, and then the Cas genes are the immune machinery. And the way these systems work is they work through a vaccination approach. So the virus comes in and essentially vaccinates that cell by parts of its DNA or its genome being added to this CRISPR memory bank. Then what happens is this memory bank is transcribed and forms these guide molecules which complex with these Cas proteins, and then these recognize and seek out the matching viral invader in the future and then target that for destruction. So if that's too complicated for you, you can think of it even more simply as essentially the bacterium storing a most wanted poster list. It's remembering, it's taking snapshots of past invaders, past threats, and then using those to recognize them and destroy them when they come into contact in the future. So we've done a lot of work over the years trying to understand this process, trying to understand how the bacteria are vaccinated by using CRISPR, and this has involved understanding the structures of these proteins as well as how these new spaces, as they're called, are acquired. And I just want to highlight as well that a lot of this research is really importantly supported by great research infrastructure that we have in the university. So we've been able to use the Center for Protein Research, we've been able to use the genomics facility as well to really develop some new techniques, particularly for sequencing these arrays. So one thing we found that was quite unexpected was that sometimes these CRISPR-Cas systems make mistakes. They have a little bit of like what we have. You can have autoimmune diseases. Bacteria also can develop autoimmunity. What happens in this case is that rather than recognizing that foreign invader and taking part of it into its genome, it in fact recognizes itself as foreign and therefore targets its own genome, chops itself up, and as you can imagine, that's pretty bad for those bacteria. They die. So that got us thinking there must be ways that bacteria can in fact control their immune mechanisms. How do they actually make sure that they're on when they're needed, but then dampened down or reduced to mitigate this risk of autoimmunity? And looking around, we realized there wasn't actually techniques available for us to be able to identify genes that control any gene of interest in a genome-wide process in bacteria. So we set out to develop a method which we called sort and seek, which I will keep it pretty simple. We essentially attach a fluorescent reporter to a gene of interest, and this could be any gene of interest in the bacterium. We make about 1 million individual mutants using transposons, and then what we can do is we can use flow cytometry to measure the amount of gene expression in every individual cell, and then we can sort these populations and then use next generation sequencing to map the genes that are influencing gene expression. And so this was really successful. It again actually involved working quite closely with the flow cytometry staff as well as Tiger Genomics facility, which helped us actually develop some slightly different methodologies than were standard. So what did we find? We actually found that bacteria have the ability to communicate with each other and control their adaptive immunity. So there was kōrero going on between these bacteria. And so much like you sitting here in this crowded lecture theatre, you could imagine that if a virus came in and one of you was infected, you'd quite rapidly spread that to your neighbors, and hopefully that's not going to happen tonight. But bacteria actually have the same issue, that if they're growing in a crowded population, if a phage comes along and infects one, it'll rapidly grow and spread to infect other bacteria that are neighboring that. So what we actually noticed was that the bacteria are using these chemical signals to communicate with each other, and they can determine when they're in this dense, high cell density population, and when they reach that population, they can change their behaviors, and in this case, the response to phage infection. So in this case, they're sensing the threat that there'd be a heightened risk of a virus spreading through that population, causing an epidemic, and that is then actually upregulating the CRISPR-Cas immune response. So even though bacteria are really simple, supposedly, they're in fact smarter than us because they are sensing that risk and they're upregulating their immune response prior to actually having that attack. So I've just told you a bunch of ways that bacteria can protect themselves from phages, and I started off by saying phages could be potentially useful to kill off bacteria. So you might be thinking this is actually not such a good strategy. But this arms race has been going on, and the phages have actually been evolving to counter some of these systems. And phages replicate faster than bacteria. They, in fact, evolve very quickly. You might have noticed I've got some little phages up the front if you haven't seen these before. And one of the great things I love about working with phages is that I'm impatient, and essentially you can see a new mutant phage evolve overnight. You can basically put it on a plate and you will see a new phage that's evaded a CRISPR defense, for example, the next day. So I think that's also fantastic for getting students excited because they can actually see evolution happening before their eyes. So phages have all sorts of ways that they can escape CRISPR and other defense systems. And Professor Horsfeld mentioned the anti-CRISPRs. So these are essentially immunosuppressant molecules that the phages produce to bind to and inhibit the CRISPR-Cas systems. So the idea is that the phage comes in, it produces this anti-CRISPR that binds and shuts down the CRISPR-Cas defense, and therefore this virus can replicate, essentially ignoring that immune mechanism. So we were really interested to try to understand different ways that phages can evade these immune systems, and we've been working in this area for a while, finding all sorts of interesting things that are happening. And so just this year, for example, we discovered that some phages actually modify their DNA in such a way that their DNA has these bulky groups on it, and this means that these CRISPR-Cas complexes that normally bind to DNA are unable to access that DNA. You're probably wondering why I've drawn these on there a little bit like lollipops, and that's because, in fact, these moieties or these modifications are in fact multiple different sugar groups that are being attached onto the surface of the DNA of these phages. So we also thought there must be other ways that phages are evading these systems. And one of the things we discovered was that there are groups of phages known as jumbo phages. So these are essentially phages that have really large genomes. They will typically have 300 or more genes per genome. And they have a really unusual life cycle. And what they do is they inject their DNA and they form a nucleus-like structure inside the cell. And this nucleus-like structure protects their DNA from these CRISPR-Cas systems being able to access it. And they also carry out the transcription within this. So one thing that this has also enabled us to do is to understand more about what kind of viruses that we could use therapeutically that might be more beneficial. And I think nothing really captures the imagination more than actually seeing this in real life. And I think this image here really beautifully shows, using confocal, that we can see the DNA of the phage in the middle of the cell, and this is a CRISPR complex that's been tagged and is unable to access that DNA. So you can really see this happening in real life. So what that has shown us that there's another type of phage which essentially has a totally unique way of evading these CRISPR-Cas systems, and therefore, if we think about this knowledge, we can now start to use phages or select phages that will potentially be able to work more effectively, because they will have anti-CRISPRs, they'll have a nucleus protecting their DNA, they might have modifications on their DNA, and therefore these will be more robust as therapeutic agents. So coming back to what we're trying to do overall, So in the phage-host interactions lab, what we're trying to do is we're trying to understand these interactions between the bacteria and their phages, and we're trying to understand these other mobile elements such as plasmids which move between different bacteria. All together, by understanding this, we're going to learn a whole lot more about microbial evolution and ecology. So just big picture questions. Phages are actually really important for global geochemical cycles as well. So there's that fundamental biology, but really what we hope to do longer term is to take this knowledge and be able to translate this into potential benefits. And so that area that we've already talked about are the phage-based antimicrobials. And the idea being that if we understand how bacteria resist phages and how we can find phages that can overcome those immune systems, then we can actually pick appropriate phages to treat certain pathogens. And so I've been really fortunate to be co-leading an MB program with Heather Hendrickson at the University of Canterbury. And what we're trying to do in this program is we're trying to take that knowledge of phage-host interactions to find and build combinations of phages that will work against important pathogens both in agriculture and in the food sector as well. And what we're also trying to do is actually build the capability within New Zealand to take a pathogen threat to then how do we find the right phages, how do we combine them, how do we scale them up, how do we formulate them, and how do we actually use these in the field. So just to touch on the one example that we're working on in this area is working on a pathogen that's causing a lot of problems within the cherry industry, and this is a Pseudomonas plant pathogen. So in fact, when you go to the orchards, you find that almost all of the plants have this disease. It's very hard to find through Central Otago plants that are not infected. Currently, the treatment is actually using copper. That's the main control mechanism, but we see that copper resistance is actually already emerging because this has been used for probably 50 years in these orchards. So what we're hoping to do is, rather than having this indiscriminate killing of bacteria using some of these agrochemicals, we're hoping to go for more precise, targeted control of the pathogens, leaving the healthy microbiome on these plants and in the soil intact. So it's a big challenge, but something which we think is worthwhile doing. And so here's just a photo of the team late last year, I believe, on a field trip when we went out to the orchards. We did some sampling of phages, but also some fruit, which was sort of a nice little byproduct. But we're going into the orchards where the pathogens are present, and we're finding phages in these orchards and then looking at their ability to control the pathogen. So just circling back to this big picture view, how do we take this knowledge and also look at other ways of applying it? And so we heard about CRISPR-Cas obviously being this really important technology. This is another area where we are trying to look at ways we can apply our findings. And so I touched on earlier that in fact studying phage-host interactions has had these quite unexpected spin-offs that have had huge biotechnological boons. So restriction enzymes, you know, the phenomenon of restriction really dates back to the 50s or the 60s, and the purification study of these enzymes in sort of the 80s led to really the molecular biology era, genome sequencing. The human genome wouldn't have been able to be sequenced without restriction enzymes, for example. So that really came from the humble beginnings of studying phages. And CRISPR-Cas, by being able to guide it with these guide sequences, we can now deliver CRISPR-Cas systems to target, bind, and cleave or modify various nucleic acid molecules. So this is a great research tool, but there are also really all sorts of success stories within the therapeutic space as well. So in this area, we've filed a few different patent applications in the last few years. So it's an area that we're really actively pursuing how we can apply some of these technologies. So that's really where I wanted to finish up, and I just wanted to thank this amazing team that I get the pleasure of working with every day. This was us recently on an excursion to Lake Tiano, and hopefully I might have convinced some of you that phages are awesome. So kia ora.
So we will hand out the medals at the end of the presentations, so don't leave. Right, I would like to introduce the next medal, which is the Rowheath Trust Award and Carl Smith Medal. This is University of Otago's top award for early career researchers. It recognises exceptional scholarly achievements within the first 10 years of a researcher's employment at the university, with a strong focus on generating new knowledge and developing innovative technology or scholarship. The purpose of this award is to highlight and reward early career researchers who are making significant intellectual contributions at a formative stage in their careers. It encourages innovation, supports the creation and sharing of new knowledge, and promotes excellence in research during the crucial early stages of one's academic development. I'd like to now welcome Professor Swatonia Green to introduce Associate Professor Andrew Reynolds, the 22nd recipient of the Rowheath Trust Award and Carl Smith Medal.
Tēnā koe Greg, tēnā koutou katoa, ko Swatonia Green tōku ingoa, he manutaki au ki te kaupeka whaiora o Te Pōti. And tēnā koe Professor Sir Jim Mann, it's lovely specifically to have you join us this evening. I'm really excited and delighted to introduce Associate Professor Andrew Reynolds from the Department of Medicine at the Faculty of Medicine, Dunedin. We're all incredibly proud of you to be awarded the Carl Smith Medal and Rowheath Trust Award. Andrew has received this honour for his globally impactful contribution to evidence-based human nutrition and public health and well-being. Andrew is a nutritional epidemiologist who focuses on describing the state of our knowledge to support achievable lifestyles and environmental change. There's a theme here with Peter, to prevent and manage the global burden of non-communicable diseases that impact us all, including diabetes, high blood pressure and obesity. Since completing his PhD at Otago only 9 years ago, Andrew's career has been striking through scholarly achievements, the generation of new knowledge, and sustained leadership in applied health research. Importantly, he does this with care, with thoughtful partnership, and a generous approach to people and his work. Basically, he's always smiling. Not only does Andrew publish in high impact journals, including first author publications in The Lancet, Nature, and Nature Medicine, his research has had impact. His work underpins multiple World Health Organization guidelines, the European Clinical Guidelines for the Dietary Management of Diabetes, American Diabetes Association guidelines, upcoming Australian guidelines, nutrient reference values, and closer to home position statements of the Heart Foundation of Aotearoa, New Zealand. It is really striking to see the actual translation and impact of Andrew's work into practice. Andrew's service to communities and people is also distinguished by his commitment to tiriti-led, equitable, and community-informed research, and also my special thanks to you to becoming the Associate Dean for Research at the Faculty of Medicine, Dunedin. Thank you, Andrew, for all you do, and I'm really looking forward to hearing your lecture, What Should I Eat? Evidence in Action. Tēnā koe, Andrew.
E haru takatoa, e taua takatahi, he taua takatini. Te pōkoko rākau, Greg, tēnā koe. Te manakura o whananga mātua mātua hoora, Megan, tēnā koe. Te manataki o te kura whaiora o tāku, Swatonia, tēnā koe. Tā Jim, tēnā koe.
Rā rakatiri rā, tēnā koutou, tēnā tātou katoa. It is so good to see people in this room. I was talking to Jim earlier today and I said, oh, I'm really worried that no one will be here. I'll be talking to an empty room. And like a good mentor, he calmed me down and he said, don't worry, it's fine. We're all coming to listen to Peter. So, and that's fair, that's fair. So, ka pai Peter, fantastic presentation and research. Okay, I have a plan. I'd like to talk to you about the team behind the research. I'd like to look at 2 research streams that have been going on for the last 10 years. And then I'd like to close in what we do next together as a team. This is the Early Career Award. I'm hoping I still have a mid and full and late career of research with Otago, so I want to tell you what happens next. Let's start with the team. What I try to express with my whakatoki is that research is done by a team. So this is not an individual award, this is an award for team. These are key players in the team. Specifically, these are Otago-based researchers where we've completed work together. So it was the assessment of this award was work done by us. However, this slide is entirely inadequate because it doesn't recognize professional research staff, such as Dr. Cherise Dana, who runs the Edgar Diabetes and Obesity Research Center, professional staff that work with us, such as through the Department of Medicine, such as Hannah, who runs the Department of Medicine, and also national, international researchers and current collaborators, either halfway through projects together that haven't been published yet or in the initial discussions of work together. It's a really big group that is taking this award tonight, and I'd like to accept it on behalf of all of us. I will keep the medal though, that one's for me, I think. So, the first research stream that I want to talk about is a dietary one, and there's 3 key sort of factors that I want to ground you in before we start talking about this nutrition topic. Number 1, what we eat is important to health. How important? Well, if you look at the global picture of mortality and morbidity, around 11% of mortality and morbidity per year is attributable to the dietary risk associated just with diabetes, heart disease, and cancers, non-communicable diseases. So it ranks highest of all of the modifiable risk factors, higher than environmental, higher than occupational, higher than metabolic. It is the top of the behavioral ones, solely due to those 3 conditions. So if what we eat is important, and working in the diabetes, heart disease, and cancer space is equally important. Every day you get exposed to nutrition messages. The majority of them are not evidence-based. They're actually there as an industry so people can make money off you consuming their content. And that could be directly advertising food that you then go buy and eat, but it can also be a bit more pernicious. Things like fad diets, wellness influencers make money off you consuming their content through the ads that they provide, and they are strong voices in this field. The evidence-based, not so much. It's a much quieter voice. And then most frustrating, the evidence-based messages that do get to the mainstream are really oversimplified to their detriment. If you were around 40 years ago, there was a big push to reduce the total fat of diets. It didn't do much health-wise. And it's really frustrating because at the time, scientists knew that, well, actually there was 4 dietary fats. 2 of them were associated with adverse health outcomes, saturated and trans. 2 of them were associated with benign or beneficial outcomes, mono and polyunsaturated fats. So that message of reducing total fat didn't do anything, but it could have easily have been reduce saturated and trans fats, replace them with mono and PUFA. So this oversimplification happens again and again, and it doesn't help people. The current oversimplification is ultra-processed foods. Yes, there are issues with processed foods and the things we eat. Yes, there's things in it that are bad for us, but the actual ultra-processed classification and the way that we look at them through NOVA is inadequate. It's ignoring the elephant in the room and focusing on some minutiae that's not important. The one that I want to talk about now, as an oversimplification that we've been working on for the last 10 years, is carbohydrates. And you would have heard about low carbohydrates versus diets versus moderate or high carbohydrates. The message that looks at total carbohydrate is not an evidence-based message. So I'll show you the data on that, and then we'll talk about how maybe we should be talking and framing carbohydrate intakes. Let's start with this. This is meta-analysis of observational data. On the x-axis down the bottom, we have the carbohydrate as a total percentage of energy, from 20% to 80%. On the y-axis, we have mortality. And what we see here is a classic U-shaped association, where the higher and lower intakes of carbohydrate are associated with greater mortality. Keto and Atkins puts you around the 20-30% of energy. Look at the disease risk associated, the mortality risk associated with that. So it's not an evidence-based message. It is observational though. If we look at trials where, say, half the people are advised to go on a low-carb diet and half the people maintain their regular diet, we don't see benefits. We don't see benefits in any clinical markers beyond 6 months, not at 9 months, not at 12 months, not at 2 years. It's not a health-driven message when you focus on the total amount of carbohydrate. And the easy reason why that's so is because carbohydrates are the most diverse of the nutrients. They range from incredibly simple things like sugars that are found on the outsides of phages to very complex things like dietary fibers. So that total look at the total amount means nothing in the context of what are the types of carbohydrate. So some of the earliest work that we did in this research stream looked at dietary fiber, the more complex things. What we did is we looked at the observational data, comparing higher versus lower intakes over decades of life, millions of person years, and we looked at randomized trials where some people got more fiber, other people didn't get more fiber. And we looked at those clinical markers most related to health: body weight, cholesterol, blood pressure, blood sugars. Before this sort of evidence synthesis, if you talk to people about fiber, people would say, oh yeah, fiber's important, it's good for feeling fuller after meals, and it's good for fecal bulking. I'm sorry, fecal bulking is not going to sell any message here. This synthesis of evidence really looked at key critical outcomes: all-cause mortality, cancer recurrence, cancer mortality, type 2 diabetes, heart disease, and stroke. And we found this consistent picture where the higher fiber consumers had less of these mortality and non-communicable diseases over the duration of the studies, decades and decades. And in the trials where people increased fiber intake, they actually lost a little body weight. They improved blood pressure, they improved cholesterol levels, and they nearly improved glycemic measures, blood sugars. This is data in the general population. It wasn't a huge surprise. It was just that this was the first time this was synthesized as a whole body, because the benefits of fiber have been supported by over 100 years of research into its chemistry, its physical properties, its physiology, and its metabolic effects. We knew that already, but seeing it in one place seemed to change things. Like I said, this is general population, but we did it in diabetes as well too. Pre-diabetes, type 1 diabetes, type 2 diabetes, gestational diabetes. And what we saw was a stronger signal than general pop. We saw greater improvements in things like body weight, blood pressure, lipids, and blood sugars, and we saw further reductions in mortality for high fiber consumers versus low. Really just to show off, we also did in existing cardiovascular disease and hypertension, and again, that same signal of benefit. All quite clear across the populations that we looked at. A couple of years later, and a little bit more exploratory, we tried to find trials that also increase both carbohydrate amount and fiber together. It's kind of hard to look at carbohydrate as an amount and as a type together, but we gave it a go. We found some trials, and all the data indicated that irrespective of the amount of carbohydrate increase, it was the fiber that drove an improvement. So high carb, high fiber, better than low carb, low fiber. It's in the title of the paper, thank goodness. And that was interesting. And that really, I think, to my mind, kicks that concept of carbohydrate as a percentage of total energy to the curb. It means nothing. You need to talk about the types of carbohydrate. That's all metadata. I should mention we also do intervention work, randomized controlled trials. In this area of increasing fiber intakes, we've done things like maximally fortifying common products like bread, putting as much fiber as possible in them to see if a single reformulation of a food product can change things in diabetes management. And also for the last couple of years, we've been delivering free healthy groceries, high in fiber, as part of a trial to over 300 people recovering from a heart attack in Canterbury, Otago, and Southland. The results from those works are just dropping now and being published, so watch that space. So that is the metadata and the trials and interventions on carbohydrate by type. We can also look at carbohydrates by source, food source. It's actually easier to talk about foods with people than it is about nutrients like carbohydrates and macronutrients and fiber. So we did that too. We looked at whole grains, beneficial. We looked at legumes, yeah, beneficial. We looked at vegetables and fruits as well too. Key sources of fiber in the diet, all beneficial. And the majority of this work all led in to inform the 2023 World Health Organization guideline for carbohydrate management. It is the evidence that underpins that, multiple different pieces. What's really interesting in this page for me is that the whole grain signal was arguably the strongest of everything we looked at. It was far stronger than the other food sources, and it was actually stronger than fibre as a standalone. A whole grain is just a grain that still has the bran intact, or as part of it. The bran contains the fibre, but also micronutrients like minerals and certain vitamins. So it had the stronger signal, so we did some intervention research over the last 10 years on whole grains as well, and we focused on whole grain processing. Because what whole grains look like in the food supply has changed over the last 40 years. Initially, they were largely intact things like oats, whole grain oats, if you eat oats for breakfast, or brown rice. But over time, the food supply has changed and they've become sort of atomized and injected into food products as a fine sort of whole grain fluff. If you go to the supermarket and you look at Cheerio cereal, it normally has a whole grain marketing ad to it because they've injected some whole grains into it. And we were really interested to see if this food processing affected how the body deals with whole grains, given that it had that stronger signal for benefit and effect. We initially started with proof of concept, very controlled environments, very set amounts of food, perfectly balanced for every macronutrient, just differing in the degree of processing, of milling of the whole grain. And we saw a consistent response in blood sugars, where the larger or the more intact the whole grain, the better or lower blood glucose response. We took that work to a free living study done in Dunedin with people living with diabetes, where we just gave them the whole grain foods, a range of 5 or 6 different products, and we said, replace the grains you normally eat with the grains we provide. And during that time, they wore CGM, continuous glucose monitors, so for that intervention duration, we watched what their blood sugars did. And sure enough, when people were having the more intact whole grains, their blood sugars were looking much better and under control. And then more recently, we've just run a trial of 160 people with type 2, again in Canterbury, Otago, Southland, where we've been delivering whole grains to their door for 12 weeks. Half of them, 80, got the minimally processed ones, the largely intact whole grains. Half of them got the finely whole grain products. And we did it for this duration because we wanted to see if we could move the most clinically relevant marker of blood glucose control, glycated hemoglobin. It's not published, but it's entirely in line with the results of the proof of concept and the shorter trial. Yes, there is actually a difference that is driven by the processing in glycated hemoglobin. Maybe no surprise, but really nice to do a sequence of research over 10 years that all show the same thing. This will have repercussions in marketing, in labeling, and probably the definition of whole grains as that data comes out, and we use it to inform dietary guidelines. So that is carbohydrate by source, and I've actually just realized I added this at the last minute. Bear with me. This is the last slide for carbohydrate, and it requires you to understand what an essential nutrient is. An essential nutrient is, well, it's in the name, it's essential to human survival. Vitamins are essential, minerals are essential, fatty acids, parts of fats are essential, and amino acids are also essential. But no carbohydrate type is considered essential in the diet. Essentiality is really important because at a policy level, they're prioritized. Essential nutrients are prioritized over non-essential. And if there's a big gap in what populations eat versus what they should be eating, that demands political action, changes to the food environment. And so no carbohydrate has been accepted as essential, but I did get notification from a journal overnight, and I haven't told Jim, so he's just learning about this now, that we had accepted this hypothesis, this argument that dietary fiber is an essential nutrient. Now, this isn't pure science and lab stuff. This is actually quite political in what we're trying to do. We're trying to elevate it above the other markers of carbohydrate because it looks like the strongest signal is the fiber content within carbohydrates. And if everyone could get on board that, if the hypothesis were true, the messaging around what we should be eating comes a lot simpler. And it can be that sort of simple message of focusing on more fiber. Congratulations, Jim. We should celebrate. Okay, so that's a nice way to end up the carbohydrate story, I think. And it was quite a linear story from where we started 10 years ago to where we are overnight. The other one is not so linear. It's a little bit more holistic because I want to talk about diabetes management as a separate topic. Some of the work I've already told you about in carbohydrate relates to diabetes, so I'm not going to go over that again. But I'm going to say when I started 10 years ago, the aim of diabetes management was actually to reduce the severity or delay the onset of complications of diabetes. That was the management strategy. And the complications are blindness, kidney failure, amputations, particularly lower leg and foot amputations, cardiovascular disease, and premature mortality. What have we been doing in the last 10 years on that, though? Well, we have done dietary stuff. These were the guidelines that Suetonia mentioned in her introduction for the Europeans. This is the European clinical guidelines, so not just carbohydrates, but the whole range of foods, dietary patterns, nutrients, lifestyle behaviors. Nice piece of work published 2023. But some of my first work in this area was actually physical activity guidelines, where we took up the physical activity guidelines for the American Diabetes Association, and we tweaked them a little bit. People did the same amount of physical activity, but we targeted it to after meals. After meals, when their body was absorbing sugars and it was going up in their system, the walking sort of knocked it down, reducing glycemic variability, peaks, and overall glycemia. Been very fortunate to work also a little bit in diabetes tech with a couple of publications, and this is primarily with Professor Ben Wheeler. Type 1s are more related to tech, but the tech is moving into the type 2 space as well too, so it's becoming more and more relevant. How a GP gives advice to their patient is a current area of research as well too, and that's largely led by Kate Campbell, PhD student on the West Coast. She's doing really interesting work there, looking at the sort of dietary advice a GP can prescribe when they're not trained at all in nutrition. And these last two are the game changers. Weight loss, mediated through lifestyle, and the new drug classes that are available for people with diabetes. And so now, I said 10 years ago, diabetes management strategies were to delay the onset and reduce the severity of chronic complications. We're now seeing in guidelines emerging that the purpose of diabetes management is to achieve diabetes remission, a removal of diabetes so that person no longer has diabetes. And this is a huge game changer for what is possible for people when they're told this. It is largely achieved through weight loss. We've done the research for the lifestyle management of weight loss here in Dunedin, working with Te Kaika Health, looking at effectiveness and acceptability. And then the drug class is probably where I want to go next in research. We're getting amazing data now that they're funded by Pharmac for people with type 2, and so we're building sort of a portfolio of evidence of what they do to people, how they interact with health inequities within New Zealand, and are there any hidden concerns? Nutrient concerns. The other concern is that the GP who administers the GLP-1RA isn't trained to give the wraparound support. So when people stop taking them, 70% of them rebound up to the same weight they were before they started taking them. So healthcare delivery and service provision are other areas where we need to figure out where these new drugs, which are fantastically effective and we can definitely use them as much as we can, fit within the broader management. Maybe it's actually dietitians who should be prescribing them because they can give the wraparound support. So lots of different things to look at there in the coming years. That's the focus diabetes management looking at. I will say, just going back to that essential concept of fibre, that is the end of that part of the carbohydrate story, but for the future years, it's also the launching pad. Because now that we've put that out there into the world, we need to look at how to defend it or refute it, be objective about it, consider what it means, and sort of champion as a possible marker of carbohydrate that isn't too oversimplistic in that health messaging world. That's all I've got time for, so thank you very much. I hope that was good, and please come to Nibbles and Drinks if you want to chat about anything.
So it just remains to really thank these two unbelievable scholars. We're so lucky to have both of them at the University of Otago, and I just hope there's some politicians online watching this just to see the global impact these researchers are having, and just extremely exciting research. So continue to ask the difficult questions. continue to seek the truth with humility, and continue to make your mark. So all that remains is to present you with the medals. So the Vice-Chancellor sends his apologies tonight. He can't be here. So luckily, we've got some members of SLT, so they've been kindly asked if they would do this. So our Deputy Vice-Chancellor Pacific, Professor Diane Sukapotonu, is here to present Peter with his Distinguished Research Medal. So Peter, please come to the stage. And I'll ask our Pro-Vice-Chancellor, Professor Megan Gibbons, to come and present Andrew Reynolds with his medal. Right, so it was fantastic to see so many people here tonight, so thank you very much for coming. And I would like to extend an invitation to the staff club. I believe that we're upstairs, and we will join you there for some refreshments. So thank you everybody for attending, and please remain behind because I think the photographer would like to get some more photos. So thank you very much.